Welcome to another episode from the Veterinary Medicine Digest. Today we are embarking on an extensive exploration into the frontier of genetic medicine as we look at a groundbreaking approach to treating a devastating condition that many of you have likely seen in your own clinics or perhaps even in your own beloved companions. We are talking about Canine Degenerative Myelopathy, a progressive and fatal neurodegenerative disorder that has long stood as one of the most challenging diagnoses in veterinary neurology. This disease primarily affects the spinal cord in older dogs, leading to a loss of motor function that begins in the pelvic limbs and slowly marches forward until the entire body is affected. It was first described over 50 years ago in aging German Shepherd dogs, and for decades our ability to intervene was heartbreakingly limited. However, the scientific landscape is shifting. We are now looking at a unique naturally occurring model for a human condition called amyotrophic lateral sclerosis, and through this connection researchers have developed a sophisticated new tool called a U1 Adaptor. This podcast will take you through the origins of our understanding of this disease, the genetic discoveries that changed everything, and the very first pilot tests of a gene silencing therapy designed specifically for dogs. To understand the future of treatment, we must first look at the foundation of the disease itself. Canine degenerative myelopathy is characterized by the progressive degeneration of the white matter in the spinal cord, eventually extending to include the lower motor neurons. The clinical signs typically emerge as pelvic limb deficits that are often asymmetric, presenting as spastic weakness and an inability to coordinate movements, which clinicians recognize as thoracolumbar myelopathy. Over a median period of about 10 months, these initial signs progress to a state where the dog can no longer walk, known as paraplegia. If the disease is allowed to continue its natural course, it eventually causes muscle atrophy, loss of reflexes, weakness in the front limbs, difficulty swallowing, and incontinence, finally leading to a complete paralysis of all four limbs and a dangerous inability to breathe properly. While the German Shepherd dog is the breed most commonly associated with this condition, it has been identified in over 90 different breeds, including Boxers, Rhodesian Ridgebacks, Chesapeake Bay Retrievers, and Pembroke Welsh Corgis. The turning point in our understanding of this disease occurred when researchers discovered that it is strongly associated with mutations in a specific gene known as superoxide dismutase 1. This was a massive discovery because mutations in this same gene are responsible for a significant portion of familial cases of amyotrophic lateral sclerosis in humans. In dogs, a particular mutation where a glutamate molecule is substituted for a lysine molecule at a specific composes when the proteins misfold and form toxic clumps inside neurons and the supporting cells called astrocytes. While this protein normally acts as a vital antioxidant enzyme, these mutations create what scientists call a toxic gain of function. This means the damage is not necessarily caused by the protein failing to do its job, but rather by the new harmful properties the misfolded protein acquires, such as disrupting the internal transport systems of the cell, causing stress to the internal cellular machinery, and triggering chronic inflammation. This shared genetic background makes canine degenerative myelopathy a spontaneous animal model for human disease, offering a rare opportunity to study a neurodegenerative condition as it naturally occurs rather than relying solely on laboratory rodents. Diagnosing this condition while a dog is still alive remains a process of exclusion. Because many aging dogs also suffer from issues like intervertebral disc disease or spinal tumors, clinicians must rule out these other possibilities before arriving at a presumptive diagnosis of degenerative myelopathy. In recent surveys, it was found that the majority of veterinary neurologists and rehabilitation professionals rely heavily on a combination of the dog's clinical history, physical findings, and genetic testing for the superoxide dismutase 1 mutation. Interestingly, while many specialists also recommend advanced imaging like magnetic resonance imaging, many owners are reluctant to pursue this due to the costs and the risks associated with anesthesia in older pets. This has led to a situation where most dogs are diagnosed based on the presence of the genetic risk factor and the absence of other obvious causes for their weakness. Historically, once a diagnosis was made, the options for management were largely palliative. No medical therapies have been shown to significantly change the inevitable progression of the disease. While various vitamins and supplements have been tried over the years, there has been no clear evidence that they provide any benefit in slowing down the degeneration. Physical rehabilitation has emerged as one of the few interventions with some documented success in supporting these patients. A small retrospective study indicated that dogs receiving intensive daily physical therapy, including gait exercises and hydrotherapy in an underwater treadmill, lived longer and remained able to walk for a greater amount of time than those who received only moderate or no therapy. In clinical practice, the goals of rehabilitation are focused on preserving muscle mass, maintaining coordination, and improving the overall well-being of the dog. Techniques like photobiomodulation, which involves applying specific wavelengths of light to the tissues, have also been explored. Some retrospective data suggests that when higher levels of light energy are delivered to the area of the spinal cord, it may help delay the onset of severe motor deficits, possibly by reducing inflammation or supporting the health of the neurons. However, even with the best supportive care, the disease remains terminal, and most owners eventually elect for euthanasia once their companions can no longer walk or begin to lose control of their bladder and bowels. This brings us to the core of our deep dive today: the development of a specific genetic therapy designed to strike at the very root of the problem. If the presence of a mutated superoxide dismutase 1 protein is what is killing the neurons, then the most logical solution is to stop the cell from making that protein in the first place. This is a process called gene silencing. One of the newest and most promising technologies in this field is the U1 Adaptor. To understand how these work, we have to look at how cells process information. When a gene is expressed, the information in the deoxyribonucleics is copied into a molecule called messenger ribonucleic acid. Before this messenger molecule can leave the nucleus and be used to build a protein, it must undergo several processing steps, one of which is the addition of a tail made of multiple adenosine molecules, a step called polyadenylation. This tail is absolutely essential for the stability and function of the messenger ribonucleic acid. The U1 adapter is a synthetic, single-stranded oligonucleotide that is designed to prevent this tail from being added to the target messenger ribonucleic acid. It works by using two distinct domains. One end of the adapter is a target domain that is custom designed to bind specifically to the messenger ribonucleic acid of the gene you want to silence, in this case canine superoxide dismutase 1. The other end is a U1 domain that binds to a naturally occurring cellular component called the U1 small nuclear ribonuclear protein. Essentially, the adapter acts as a tether, bringing this cellular machinery to the specific gene transcript and blocking its ability to receive that essential adenosine tail. Without the tail, the transcript is unstable and is quickly degraded by the cell, which means the toxic protein is never produced. This technology is particularly exciting because it has shown very high potency and low toxicity in earlier studies, even outperforming some other gene silencing methods in laboratory models. A pilot study was recently conducted to test whether this U1 adapter technology could be safely and effectively used in dogs. This research was performed in two distinct phases to prioritize safety and determine the correct dosage. The first phase involved 7 healthy adult dogs and 1 dog that was already in the late stages of degenerative myelopathy. These dogs received a single dose of the U1 adapter designed to target canine superoxide dismutase 1, administered through an injection into the fluid surrounding the spinal cord, which is known as an intrathecal injection. The goal was to see if the treatment could actually reduce the levels of the target messenger ribonucleic acid in the central nervous system. The results were very encouraging. In the dogs that received the treatment, researchers found that the expression of superoxide dismutase 1 ribonucleic acid in the spinal cord was reduced to a median of only 37% of normal levels. This means the treatment successfully achieved a silencing effect of over 60% near the site of injection. The second phase of the study focused on 4 dogs that were in the early stages of degenerative myelopathy. These dogs received monthly intrathecal injections of the U1 adapter for as long as 10 months. This was a crucial test of how well the dogs could tolerate repeated treatments over a long period. Clinical evaluations, including physical and neurological exams, blood tests, and analysis of the spinal fluid, were performed regularly to watch for any adverse reactions. The researchers found that the dogs tolerated the procedures and the genetic agent remarkably well. There were no signs of clinical or microscopic toxicity in any of the organs examined. Only 2 episodes of temporary hemodynamic instability were noted during the 34 total injections performed, which were managed successfully. Importantly, the blood tests and spinal fluid analysis remained stable throughout the study, indicating that the long-term administration was safe. While the study was primarily designed to assess safety rather than to prove clinical efficacy, the observations of the diseased dogs provided important insights. The researchers noted that despite the successful silencing of the gene, the clinical decline of the 4 affected dogs continued along a path that was consistent with the known natural history of the disease. The time it took for these dogs to lose their ability to walk was very similar to what has been observed in untreated dogs. This lack of a clear clinical improvement, despite the reduction in the toxic gene's expression, points to several important challenges that still need to be overcome. One of the primary issues is the distribution of the treatment. Because the drug was injected into the lower part of the spinal cord, the researchers found a decreasing gradient of the medication as they looked further up toward the brain. While the silencing was strong near the injection site, it was much less effective in the brainstem and the brain itself. As degenerative myelopathy progresses, it eventually involves the entire spinal cord and parts of the brain, so achieving a more uniform distribution of the therapy is going to be essential for future success. Another major hurdle is the timing of the intervention. By the time a dog begins showing clinical signs like weakness or ataxia, a significant amount of damage to the neurons has already occurred. In human clinical trials for similar conditions, it has become increasingly clear that treatment should be initiated as early as possible, ideally even before symptoms appear in those known to carry the genetic mutation. The pilot study in dogs allowed for the inclusion of animals that were already showing signs, which might have limited the therapy's ability to halt the progression of the damage. Future research will likely focus on refining how the drug is delivered. The researchers mentioned that using a version of the drug that can cross the blood-brain barrier after being given through a standard intravenous injection would be a major step forward, as it would avoid the need for repeated general anesthesia and provide a much more consistent level of the drug throughout the entire central nervous system. In addition to genetic silencing, researchers are looking at other ways to protect the neurons from the toxic environment created by the superoxide dismutase 1 mutation. For example, studies have shown that the loss of a specific glutamate transporter in the spinal cord may contribute to the death of motor neurons in both dogs with degenerative myelopathy and humans with amyotrophic lateral sclerosis. When these transporters fail to remove excess glutamate from the space between neurons, it can lead to what is called excitotoxicity, essentially overstimulating the cells until they die. Finding ways to restore these transporters or using other neuroprotective substances, such as curcumin, which has shown some potential in slowing disease progression in small studies, might eventually be used in combination with gene silencing to provide a multi-pronged approach to treatment. Ultimately, this research provides a vital foundation for the future. We have seen that gene silencing using U1 Adaptor technology is a feasible and safe approach in a large animal with a naturally occurring disease. This is a significant milestone as it moves the field beyond laboratory rodents and into a more complex and relevant clinical setting. The similarities between canine degenerative myelopathy and human amyotrophic lateral sclerosis mean that every discovery we make in our veterinary patients could have profound implications for human medicine as well. While a cure for this devastating condition remains on the horizon, the ability to safely reduce the expression of the gene responsible for the damage is a monumental step forward. It brings us closer to a day when we can offer more than just palliative care, a day when we might finally be able to change the trajectory of this disease for our patients and their families. As we conclude this deep dive, it is clear that the path to a cure is complex and filled with biological challenges, but the dedication of the researchers and the participation of dedicated pet owners are driving the field forward at a remarkable pace. We have explored the intricate mechanisms of the spinal cord, the dark secrets of misfolded proteins, and the elegant logic of ribonucleic acid interference. These are the tools that will define the next generation of veterinary medicine, where we treat diseases not just at the level of symptoms, but at the level of the very code of life itself. Thank you for listening. The titles for the reference papers are in the video description. From VetMed Digest, where we keep minds inspired, hearts light, and tails wagging.